Hello, hi. We'll continue our biotechnology course with the basics of gene expression and basics of gene expression control. So, gene expression, generally it's a production of messenger RNA by a cell. All cells of course, all cells of organism contain the same genome. So how and why are skin cells different from brain cells or liver cells? Because cells can regulate or control the gene they express. The gene regulation is how genes turn on and off in response of different signals to different signals. Transcriptional co-regulation. This is control the amount of messenger RNA transcribed from a particular gene. RNA polymerase cannot bind to promoter region without the presence of transcription factors. Enhancers. These are sequences bind to regulatory proteins called activators, while repressors are sequences can decrease transcription. If you look closely here, you'll see a DNA sequence that have activators like hormones, receptor proteins that can bind to enhancer sequence, specific sequence upstream of the gene. If you go downstream of the gene, just before the gene, you'll see the promoter sequence that have something called cat box or TATA box, TAT box, the famous part of promoter region. This is followed immediately with the start site of gene expression. Here the activator protein binds to the enhancer sequence in the DNA. This brings the bound activator close to the promoter region, and the other transcription factor and RNA polymerase come and try to assemble to start the process of transcription. Once they are assembled at the site of the promoter, the RNA polymerase with the associated transcription factors and the activator protein start to transcribe the gene, and RNA synthesis is formed. Bacteria have operones, and these operones regulate gene expression. Operones are clusters of related genes located together and controlled by a single promoter, which differs from the eukaryotes, which can have a multiple promoter. The operator is the region within the promoter that regulates gene expression in response to nutrient requirements. We'll take an example as a lac operon. The lac operon controls the formation of the messenger RNA of three enzymes which are necessary for transport and breakdown of lactose. If you can see, the three genes are Z, Y, and A. This is the genes, structural genes. And the RNA polymerase is located on the promoter region, which is followed by the operator region, which is in red. Okay, this is a DNA sequence. So the lactose is an operator activator. So actually the lactose, the molecule of lactose, it is an operator activator which switches on the lac operon. If no lactose found, lac repressor bind to the lac operator and blocks the RNA polymerase binding and hence blocks transcription. In other words, the lac repressor once bind to the red region, the lac operator will block the RNA polymerase from moving on the gene to transcribe. Lac operin consists, as we said, of three genes: lac Z for galactase, lac Y for permease, lac A for acetylase. Again, the lac operon, if you see the mechanism, the repressor protein is encoded from the I region, the inhibitor region, which lead to forming a repressor protein. This repressor protein, once there is lactose in the gross medium of the bacteria, will bind to this repressor protein, and it will prevent its binding to the operator region. Hence messenger RNA is transcribed, as we see, and it have a long messenger RNA that is transcribed, only one messenger RNA that is translated into three different proteins. And that's a major difference between prokaryotes and eukaryotes, because there is three genes is transcribed under the control of one promoter, while in eukaryotes we will have the three genes with different promoters. How is messenger RNA read? Actually, the genetic code. It's a universal genetic code used by all living organisms, and as you know, it works in three nucleotide units called codons. Each codons codes for single amino acids. There is as well one start codon and three stop codons. You can see from the table. As you can see from the table, you can see as well that one amino acid may be coded by more than one codon. The messenger RNA have possible 64 possible codons. It's actually the combination of 4 times 4 times 4. This is a universal code that specifies amino acid. 61 codons for amino acid and 3 codons for the stop codons. Only 2, methionine and tryptophan, have a single codon, while leucine, serine, and arginine have 6 different codons. The synthesis of proteins and prokaryotes must begin with methionine. The initiator codon is AUG. Some other codons like GUG and CUG are rarely or sometimes used as initiator codon in some eukaryotes or few bacterial messenger RNA. What is reading frame? Actually, reading frame is a sequence of codons that start from a specific start codon to stop codon. Messenger RNA can be transcribed into 3 different frames, but most messenger RNA can be read only in one frame because the stop codon is encountered in other 2 possible frames and terminate the translation before the functional protein is produced. To have a more illustration, this is frame 1 of translation. So messenger RNA is translated into frame codon by codon by codon. In frame 2, there is a shift of the first nucleotide is ignored in the process of translation. So you'll have a shifting. That's why it's called frame 2. So the codes have been changed and different protein, different polypeptide is formed. As well as in frame 3, when the first 2 nucleotides of the messenger RNA are ignored and another protein is formed. Normally the messenger RNA is read on only one frame, and normally it's a frame 1. If you look closely here to the table, so this first column represents the first position while reading in the process of translation, while this second position of the triple code represent these 4 nucleotides, and this is the third position. So this is the 3 position. To have an example, if you have a DNA that have this sequence, and you want translated, you want to transcribe it into messenger RNA, this messenger RNA will be transcribed in this way. Keep in mind that we always read the nucleic acid from the direction 5-3-. So actually TAA can be transcribed into AUU, but we needed to write it in the correct orientation from 5- to 3-. Okay, again, TAA, the first codon in the DNA, TAA is transcribed into AUU. And this is the 3' end. Okay, so normally we write the nucleic acid from the 5' ends. If you want to translate this, to make it, why you normally write it in the 5' end? Because it's easier to understand translation. So you have the first codon now is GCU, and this gives alanine amino acid, and you can see this alanine. And if you try to exercise on the other 2 codons, you'll find that they are translated into cysteine and leucine. The levels of gene expression. So the gene regulation is on different levels. It's not only on the level of the nucleus or the level of cytoplasm or the level of the translation. You have the first level, the regulation of gene expression while the DNA got unpackaging, and due to the involving DNA demethylation and histone acetylation, regulate if the gene is available for transcription, and this is the first level of regulation. While the second level is the transcription regulation. This means to transcribe or not transcribing the gene. The third level, the RNA processing, which I've seen it consists of 3 different processes, the capping and the polyadenine tail and the splicing of entrons and removal of entrons. The next level is the transport of the cytoplasm. It's a mode of regulation of gene expression as well. The next level is the translational regulation and degradation of messenger RNA, while this is followed after the protein is translated into post-translational regulation, either by chemical cleavage, modification, transport to cellular destination, where the function of the protein is active. After the protein have been fully formed and active and done its function, it must be degraded for recyclation of the amino acids. If we continue of the level of gene regulation, we will find another category of RNA, which are non-coding RNAs, which its major type is microRNAs. Recently, microRNAs have been a lot of microRNAs have been discovered, and they found that these non-codings nucleic acids can silence genes, reveals another level of gene expression regulation. You can see that the microRNAs are transcribed from the DNA as a pre-messenger RNA, microRNA. Under the effect of DROSHA enzyme, it will convert it into pre-microRNA. There is some processing a DROSHA inside the nucleus. Following the export of this pre-microRNA into the cytoplasm, under the effect of Dicer, there is a cleavage and the microRNA is formed. So let's try to explain in more details. So the microRNA genes transcribe a primary transcript called the pre-microRNA that folds into hairpin loop similar to the folding pattern of a transfer RNA molecules. This enzyme Dicer cleaves this pre-microRNA into single-stranded microRNA that are around 21 to 22 nucleotides. The microRNA bind to a cluster of proteins to form a ribonucleoprotein complex. This microRNA complex can bind to messenger RNA by complementary base pair, and this microRNA either bind and silence gene expression by blocking translation or by targeting the bound messenger RNA for degradation by enzyme in the cells. So it's actually another mode of regulation at the level of translation. The next subtopic is mutation, causes and consequences. Of course, mutation is a change in nucleotide sequence of DNA. It's a major cause of genetic diversity, can also be harmful. Types of mutation either point, silent, missense, nonsense, frameshift mutation. This is inherited through gametes or acquired genomes of the somatic cells. Human genomes are approximately 99.9% identical. There is only 0.1% difference between individuals, roughly 3 million difference between different individuals. Most mutations have no obvious effects. Other mutations strongly influence cell function, behavior, susceptibility to genetic diseases. Gene mutations can cause changes in protein structure, function, synthesis of non-functional proteins, or no protein synthesis can lead to a change or loss of trait or a phenotype. So biotechnology is being used to detect and correct mutations. If we look too closely, this is one of the important slides. If you look closely to the types of mutation, you'll see, if you assume that this is a normal wild type genes, have two double-strand DNA, a single-stranded DNA have been transcribed into messenger RNA. Okay, so we have a messenger RNA that's been translated into a polypeptide formed of 4 amino acids: methionine, lysine, phenylalanine, and glycine, followed by a stop codon, so there is no amino acids. And this is the normal behavior of gene transcribed into messenger RNAs and followed by translation. There are generally two types of mutation, either base pair substitution or base pair insertion or deletion. You will see that there is a silent mutation in which the nucleotide A is mutated from the original, because originally it was G if you look to the wild type. So this gives another nucleotide when transcribed that doesn't affect the amino acid sequence of the translated protein. That's why it's called silence, no effect on the amino acids. The other type is missense change codon. So you will see that T have been changed, leading to the change of the amino acid that has been inserted in the polypeptide. So that one is a different proteins. The other type is nonsense mutation, creates a stop codon. So the mutation in the DNA leads to formation of a codon that lead to a stop codon. That means the protein formed is truncated only of methionine. Okay. The other category of mutations, the frame shift, means the frame shift. That means that you will see that the T nucleotide have been lost. So the codon start to read in the absence of T after the transcription. So two new amino acids have been inserted, which results in a protein or polypeptide that totally different from the wild type. While in frame shift causing immediate nonsense, This means insertion of the nucleotide A leads to formation of a stop codon. That means formation of a truncated version of the protein, as well as insertion of deletion of 3 nucleotide will affect the framing of the translation. So the reading frame will be distorted. So another amino acid will be inserted because of the disappearance or the addition of 3 nucleotides. The last subtopic in this lecture is revealing the epigenome. So epigenome refers to the modification in chromatin structure which do not involve mutation DNA sequences. So there is a change in chromatin structure and no mutation is involved. Some epigenetic modifications are inherited, others vary among generations. Some these of modification can be reversible, while others are long-lasting and irreversible. Epigenetic changes can differ between cell types in the body and in normal and diseased tissues. Diet and environmental conditions can influence the epigenome. It is now known that the epigenetics play a major role in clarifying how patterns of gene expression can vary during embryonic development. So you can see here that the chromatin involved in epigenetics, you can see that DNA methylation, histone modification, remodeling complexes, histone variants, non-coding RNAs. All of them control when a gene transcribed and how much of messenger RNA are transcribed, when it's switched on, when it's switched off. So it's all types of modification that have a level that can regulate gene expression without mutation, or can just change the protein without the protein pattern without mutation. And in next lecture, we'll deal with recombinant DNA technology and genome.